And Dr. Parmanand Verma is the drug inspector at Chhattisgarh, sir. He is our All India National Treasurer and Webinar Committee Chairman, sir. Can you come?
Oh, okay.
Parmanand?
Yeah, I've been seeing his name on the chart.
Recently.
Mr. Parmanand? Mr. Parmanand?
Hello, sir, I am audible?
Yes,
sir.
Yes.
Audible.
Yes, good evening.
Yes.
Good evening, sir.
Yeah, you are audible and also visible now.
Yes, sir.
Nice meeting with you.
Yes, thank you, sir.
Great.
We are eagerly waiting for you, sir, to hear you.
Yeah.
Thank you.
I think we are close to the time, and can we start?
Yes, sir.
Yeah. One more minute.
Sure.
We created with 5 members committee for this webinar, sir, under the supervision of Parmanand Verma, Krishna, and Mr. Taneja, also former drug controller from Haryana, and Miss Mahalakshmi, drug inspector from Kerala, and Ahuja, sir.
I see.
So every month
Very
we
nice.
are conducting, sir. This is the 10th webinar, sir.
Wonderful. Yeah.
Yes, sir. Ahuja, sir, start. Ahuja, sir.
Largest DW and very hardworking leader, Shri Koteshwar Rao. I am DRK sir, and I also welcome Dr. Kalai Selvan and especially congratulate him for inauguration of IP 2026, a fantastic document addressing many problems. He will discuss in length and he will definitely answer many questions in the minds of the drug regulators, not taking much because both the speakers, our honorable guests, ocean of knowledge, and we want to hear them. I welcome all the participants, both the DCOIWA webinar committee. I welcome all of them on behalf of DCOIWA. Thank you, and let we begin the webinar. Thank you very much.
Thank you, sir. And today our chief guest will be
Sir.
Dr. Kalai Selvan. And before giving his chief guest lecture, I request Mr. Krishna to read the introduction of Dr. Kalai Selvan. Mr. Krishna.
Good evening, everyone. It is very glad to welcome you all by DCOIWA for the 10th webinar series held by the DCOIWA in coordination with the IPGA Telangana. So I welcome Dr. Selvan as chief guest for this webinar, and Dr. Selvan pursued his B Pharma and M Pharmacy and PhD from University of Delhi. Dr. Selvan currently as the scientific secretary cum scientific director of the Indian Pharmacopoeia Commission at Ghaziabad, which is under Ministry of Health and Family Welfare, Government of India. Dr. Selvan has been associated with IPC since 2009, contributing significantly to the domains of pharmacopoeial standards, rational use of medicines, drug safety, and regulatory systems. He played key role in the conceptualization and nationwide implementation of the Pharmacovigilance Program of India and the Materiovigilance Program of the India, which have strengthened the Indian surveillance framework for the safety of medicines and medical devices. Dr. Selvan has represented IPC on various national and international platforms, including WHO, and continues and contribute to global invitees for strengthening the pharmacovigilance system in low and middle income countries. In recognition of his work, the WHO headquarters in Geneva acknowledged him as one of the significant contributors to global pharmacovigilance in their 50th celebratory album of the International Drug Monitoring Program. He has authored 11 chapters and published over 130 research and review articles in national and international journals. He also a recipient of prestigious fellowships awarded by the Department of Science and Technology and the All India Council for Technical Education. So I welcome Dr. Selvan to address this webinar.
Thank you, Krishna, and over to Dr. Kalai Selvan, please.
Thank you. Good evening and very happy new year to all of you. Respected Narendra Ahujaji, Dr. Koteshwar Rao, other dignitaries, delegates, speakers, very good evening and morning as well. So I came to know some of the experts and delegates also joining from ES. So let me also say best wishes to all of them. Very good morning and good evening to all of you. It's a great opportunity for me. The organizers invited me at the right time, and it's a great opportunity while still we are celebrating and enjoying the new year. I am also happy to share with you that yesterday we had a landmark achievement, landmark event, as mentioned by Dr. Narendra Ahuja. We released IP, Indian Pharmacopoeia 2026. So to begin, before begin my address, let me thank the Drug Control Officers Welfare Association and IPGA Telangana branch for inviting me and making me a part of this webinar. Once again, my best wishes and New Year greetings to all of you. Really, the webinar today, the topic, what they have decided, what they have selected a topic is like drug interaction. This is what the topic which selected for the organizers, by the organizers, and it's a really need of the hour actually when it comes to safety or medication error, etc. So I'm sure this drug interaction and deliberations and discussions will definitely going to help all the participants, those who have joined today's webinar, will go a long way. So if you look at, there are emerging number of emerging problems, like let it be in pharmacovigilance. There also drug interactions are also going to be an important role because of drug interaction. Many adverse events we would have prevented. Those, all of them would have been prevented. So that the basic root cause is either medication error or drug interaction or lack of efficacy, etc. So it's a quite unique and the need of the hour to discuss this topic, and will try to contribute for the patient safety. When it comes to this is one aspect of safety, but quality also directly linked with the patient safety or quality related or safety related part. So the quality is one of the important area where we need to address, and we need to look at the quality of pharmaceuticals. Yesterday we had Indian Pharmacopoeia come out of 10th edition of Indian Pharmacopoeia, which was released yesterday by the Honorable Union Minister Sri J.P. Nadda ji along with other dignitaries. So this is going to be a milestone and going to make a lot of impact, strengthening the quality standards and quality system of medicines. And it's a right opportunity, as most of the regulators, senior regulators are present over here. I am sure with all your support and guidance, let us implement this effectively. Whatever the standards we developed and we published in an IP, but the regulators' role plays a very important. It's going to be very, very important role in terms of implementation. So that is the reason in the upcoming months, upcoming days, we are going to have huge series of webinars and um, training programs for the stakeholders. And I think after launching the IP Indian Pharmacopoeia, and this is my first official engagement, and it's a very important engagement, I would say today. What I'm trying to convey to address the gathering. So this IP 2026 will be effective from 1st July, but we have given sufficient time to the manufacturers to adapt or to implement, to set up the facilities as per the new requirement of IP 2026. So this is one of the background about Indian Pharmacopoeia. And next one, if you ask me the saline futures, I mean major changes what we brought in the new edition. So there are 21 new drugs and pharmaceuticals monograph. New standards have been developed. Out of 121, there are 21 monographs, 21 drug standards, which are not available in any other pharmacopoeia. First time, it is coming in Indian Pharmacopoeia under the chemical category. And the remaining other monographs have been, 90 monographs have been, new monographs have been introduced. 100 remaining monographs. Out of 121, 21 of them are new, first time coming in IP, and the remaining hundreds are maybe available in other pharmacopoeia, but it's new for Indian Pharmacopoeia this time. Plus 400 plus more than 400 monographs or standards have been upgraded with latest technology. And in order to estimate or ensure the elemental impurities, maybe mercury or cadmium, nickel, all those elemental impurities shall be required to be estimated or shall require to be ensured by ICP-MS method. So there are certain new salient features, and there are biotechnology derived monographs, biosimilars. Now the Indian Pharmacopoeia Commission is also working towards developing the standards for biosimilars. So even the total number of standards or monographs are available for biosimilars or biotechnology derived products. We are at par with any other pharmacopoeia. As compared to other pharmacopoeia, we are also leading in the biosimilar and biotechnology derived monographs. And then there are new additions, many new additions to specify. Even in the yesterday inaugural session also, Honorable Minister highlighted that there are 20 blood components monograph. Huge impact. It is going to have a huge impact and very significant milestone. We have taken that steps we have taken to ensure the quality of blood and blood products used in thalassemia, anemia, dengue, and many other cancer and many other diseases. So these 20 blood components monographs are its standards will be helpful to all the transfusion medicine department, while going for a transfusion therapy or blood component therapy. IPC also, this new edition also supports the Government of India's mission or Ministry of Health and Family Welfare's mission on Anemia Mukt Bharat, in which we have introduced ferric carboxymaltose injection first time in IP, and it is available only in IP, not in any other global pharmacopoeia. Again, the blood components monograph also first time in IP, and it's not available in any other pharmacopoeia. So like this, there are many saline features and significant steps we have taken to strengthen the pharmaceuticals drugs quality and standards. So I request all the delegates and participants kindly update the IP and keep in touch with Indian Pharmacopoeia Commission for its effective implementation and ensuring the safety and quality of medicines. Well, this is one way of quality part. Now let me come to safety. Quality and safety both are two different sides of a coin. Safety of pharmaceuticals are also a part of responsibility of Indian Pharmacopoeia Commission to ensure the safety. As the Indian Pharmacopoeia Commission manages or coordinates the pharmacovigilance program of India, a team under the Ministry of Health, on the government of India to monitor the safety of medicines across India. So again, we have done a lot of work in pharmacovigilance. It's an active, very active scheme, and it's for the regulatory system to take any regulatory decision based on our own data. There was a time before 2014, since I was a part of I'm part of Indian Pharmacopoeia Commission since 2009. There was a time when I joined, there was no data available in our system. I mean, there were no data based on our own population data. So whenever the regulators asks, say IPC for pharmacovigilance data or patient safety data, we were not having that time. Either we used to ask some other country to help us to provide the data to take a decision, which is it's irrational. It's not at all scientifically it is not correct actually. We have different population, we have different kind of ethnics ratio, etc. So now the situation is different. We the whole entire world depending on Indian data, whether WHO or anyone, any other international organizations or countries, they depending on our data, we would able to generate for our own population, as well as we are also supporting other countries for regulatory decisions. So this is the current status, but still we have to go a long way. Still the scheme needs to be reached to the public and nook and corner of the country. Every citizen should aware of pharmacovigilance. Then only we can able to say that the program is a complete success. So it is a huge responsibility. I urge all the stakeholders, those who are present here, you support the pharmacovigilance also. You try to contribute and try to disseminate this information to all the stakeholders, and let us make this as a robust and citizen or patient centric program. Same way like pharmacovigilance, we have a material vigilance to ensure the safety of medical devices. So both pharmacovigilance and material vigilance should work together, because when the medicine, before giving the medicine, the patient undergoes different diagnostic procedures. So there also devices are involved. So the diagnosis, then only treatment. So the diagnostic devices are medical device. So we have to ensure the safety from there, then medicines. So these are the couple of things which I thought of sharing with you. And when you discuss the drug interaction, the Indian Pharmacopoeia through National Formulary of India also address this issue. So again, the National Formulary of India is also a reference document for promoting the rational use, including addressing the drug interactions. So I'll be happy to share with you the next edition of NFA, which is again going to be launched very soon, maybe in another couple of months. The National Formulary also will be available to the pharmacists and other prescribers, practitioners for promoting the rational use. I think that's all from my end. I think let me take this opportunity to thank all of you, and thanks for this opportunity. And I wish the webinar grand success. Thank you so much. Once again, I congratulate Drug Control Officers Welfare Association for inviting me. Thank you so much.
Thank you very much, Dr. Kalai Selvan sir. First of all, thank you very much for accepting our invitation, though you are very busy till yesterday night in inauguration of that Pharmacopoeia IP 2026. And thank you very much for your valuable information which you have given. And in future, we are going to conduct one webinar exclusive on Pharmacopoeia with the help of you. Thank you very much, sir.
I think that will be great. That's a great suggestion, sir. We can have an exclusive webinar only on Indian Pharmacopoeia. So yes. I will be happy to contribute. I can bring all the experts related to blood products, reference standards, etc. So please let me know. I'm fully I'm ready to help you whatever the way you want to conduct the webinar. Thank you.
Thank you, sir. Thank you. And coming to today's keynote speech, Drug Interactions, a Regulatory Perspective by Professor Krishna R. Devarakonda. We generally always call him as a DRK, sir. He is one of the leading pharmacists in India and abroad also. And I personally invited him in Houston, America in my last visit for this webinar, and luckily he came to India. Now he is in Hyderabad, and today he is in front of us. So before handing over the screen to DRK sir, I want to give brief introduction of DRK sir. And before that, I am happy to say that the participants include the classmates of Dr. DRK, Sri Tata Rao sir and Sri Ashok Rapaka sir, and his colleague Professor Achaya sir, and many IMA doctors are there from IMA, and there are drug regulators, the pharmacy, PharmD students, IPGA, IPA, and APTI people, and many of watching through YouTube also. Today there is Dr. Jaga Shetty sir, former drug controller of Karnataka. So welcome to all you sir. And let me introduce Dr. Professor Krishna R. Devarakonda is M.Pharm PhD FCP AvH Fellow, pharmaceutical scientist, clinical pharmacology leader, educator and mentor. He is from Richmond, Texas, USA. Professional summary: passionate educator and globally experienced pharmaceutical scientist with 30 years in academic and research and 18 plus years in pharmaceutical industry. Renowned expert in clinical pharmacology, model-based drug development, and NDA, ANDA strategy with a strong record of leadership in drug discovery, clinical development, and regulatory science. Successfully mentored 25 plus PhD scholars and 60 plus M.Pharm students. Current appointments are Founder and Chief Scientific Officer, 6S Pharma Incorporation, New Jersey, from 2024 and present. Senior Scientific Advisor, Cironex LLC, Massachusetts, from 2025 and present. Senior Scientific Advisor, Isha Therapeutics LLC, New Jersey, 2023 up to present. Senior Pharmacology Research Advisor and Co-Principal Investigator, Daya Drug Discoveries LLC, Missouri, from 2024 onwards. Adjunct and Visiting Professor of Pharmaceutical Sciences, University of the Pacific, California, from 2008 to present. Prior leadership and industry roles: Vice President Clinical Development, Mallinckrodt Pharmaceuticals, Missouri, from 2021 to 23. President and CEO, ARK Scientific LLC, Missouri, from 2018 to 2021. Senior Director / Head Clinical Pharmacology, Mallinckrodt Incorporation, New Jersey, from 2016 to 2018. Director Clinical Pharmacology, Mallinckrodt, Missouri, from 2010 to 2016. And Clinical Pharmacology Fellow, Covidian, Missouri, 2007 to 2010. Academic career: Professor of Pharmacology and Clinical Pharmacy, Kakatiya University, Warangal, India, which is my native place, from 1999 to 2007. Associate Assistant Professor of Pharmacology, Kakatiya University, from 1979 to 1999. Assistant Professor of Clinical Pharmacology, Nizam's Institute of Medical Science, Hyderabad, from 1991 to 93. His expertise: Dr. D.R. Krishna is expertise in clinical pharmacology and translation medicine, PFD modeling and simulation, model-based drug development, MBDD, NDA/ANDA strategy and regulatory submissions, precision medicine and pharmacogenomics, drug discovery and clinical development leadership. Patents and scholarship. Patents 10 plus. Publications, books, chapters, invited talks, abstract more than 200 plus. Google Scholar publication profile. And honors and awards: Fellow, American College of Clinical Pharmacology, FCP, Professor M.L. Khurana Memorial Award during IPC 2016, and Mallinckrodt Innovation Award in 2014, and COVID in Innovation. and many are there. Actually, I condensed it after
Thank you
condensation.
so
Also, it
much.
has been improved. And over to Dr. DRK to start his presentation. There is no time bar, sir. You have leisure time, and after that, there may be questions also. Thank you. Thank you, sir.
Thank you so much, Mr. Koteshwara, for a very liberal introduction about me, and Dr. Kalai Selvan for your excellent introductory remarks related to most recent developments regarding Indian pharmacopoeia. I'm really pleased to know that Indian pharmacopoeia has been moving on, developing leaps and bounds. It's really a great thing for India. And the developmental program that you envisaged, I think it's really something noteworthy, and many countries throughout the world should follow this suit. And coming back to today's topic, drug-drug interactions, a clinical and regulatory perspective. This is a topic that Mr. Koteshwar has asked me to focus on, and I'm really happy to do that. The only thing is that since the cross section of the audience today is so broad, we have probably fresh pharmacy graduates all the way through people who have been in academia, industry, regulatory science, with enormous amount of experience and knowledge. So probably it's a big challenge for me to be able to cover, to satisfy the palate of each and every individual. Nevertheless, I will try to make a humble attempt, trying to touch the basic concepts and then try to keep on adding more and more, build up a small structure, and try not to remember everything that you are going to see here or hear from a talk. Just try to grasp what exactly I'm trying to tell, so that when you go back and see in future, you should be able to understand the significance of drug interactions, as well as the impact of drug interactions, as Dr. Kalai Selvan mentioned, in the adverse drug reactions, which has been one of the most important problems related to the safety. And as you know, regardless of which regulatory agency we are talking about, whether it is US FDA or European EMA or Indian DCGI, the fundamental goal is to protect the patient, that is protecting the safety of the individual. Then comes the question of efficacy. So with that, I will just define what a drug interaction is. There is some difference between drug interaction and drug interaction. Drug interaction is an offshoot of drug interaction. So drug interactions are loosely, these are the ones which occur when the effects of one drug are changed by the presence of another. So you are considering two drugs, either they are administered concomitantly or probably one after another with a time gap. Nevertheless, if there is any interaction between the two, or at times you may be administering a herbal medicine, some kind of herbal drug. For example, in India, people use a lot of alternative herbal products like ashwagandha and chavanapras and so many other things. So we don't know what kind of herbs that it contains, and each and every herb has got several chemical constituents present in it. And so there is a possibility, there is a potential for those ingredients to interact with the drug, the main drug that you are administering. Likewise, there can be interactions between foods and drugs, drinks and drugs, and any other environmental chemical agents that are going into the biological system. So any kind of interaction between these two can be called drug interactions. And the major focus of my today's talk is not between drug and food, drug and herbal medicines, drug and other things, but mainly drug and drug. So for that, I have decided to touch upon some of the basic concepts, because as there are a lot of students or probably fresh graduates, I thought it's better to touch upon some of the basic things So I think all of you must have had some kind of training or understanding about pharmacology and also a little bit of toxicology. So both of them are pretty much related. When we are talking about the drug action, which is clinically required, then probably we are in the pharmacology space. And if these drugs produced when the dose is exceeded or otherwise, any situations, adverse reactions, and so on, then we are talking about toxicology. So you know pretty well that perhaps many of you must have read in very early classes of pharmacology that every drug is a poison, and what decides is the dose. So when you are dealing well within the therapeutic range, then it is useful, and when you are exceeding that, it could cause toxicity. Nevertheless, these molecules, all drugs are chemicals, and these chemicals are also xenobiotics, that is, they are foreign to the body, and so the body always tries to get rid of them. And as long as the drug molecule is within the system, it is going to produce the desired pharmacological response, and for that, it has to bind with what we call drug targets. The drug targets are generally some kind of protein molecules or nucleic acids or any kind of material that is typically associated either with the cell membrane or the cytoplasm of the cell. And these can be called as receptors, ion channels, and enzymes. These are normally considered drug targets. And there are also transport systems, and you have got nuclear receptors which are present within the cell. And then if you go even further down, you have DNA. So drug molecules, they reach these target sites, bind there. Normally the binding is transient. It is for a short period of time. And then they get dissociated and they are gotten rid of. And the most important medium that takes the drug molecule is plasma or blood. It takes to different tissues. And so the drug enters the system upon administration, and it crosses several barriers and reaches the target sites and produces its pharmacological effect. So that is what pharmacology is typically called as. So pharmacology can be further divided into two important branches. One is pharmacokinetics, the other one is pharmacodynamics. I think many of you know what these two are. Pharmacokinetics is something which is loosely called as the fate of the drug. So what is fate of the drug? That means what your body does to the drug molecule. And or you can call it as the time course of certain processes like absorption, distribution, metabolism, and excretion. So since we are using the word kinetics, meaning related to time, so what happens to the drug for a period of time until it, starting from the point of its entry into the system until it is removed from the system. And during this period, the drug molecule tries to reach different parts of the body thanks to the circulation, and it reaches the target sites, and then it interacts with the target sites. And this kind of interaction can be quantified, and that is within the purview of what is called as pharmacodynamics. And when you are talking about these two sciences, pharmacokinetics and pharmacodynamics, that is what the body does to the drug and what the drug does to the body, in the context of a patient, then it is called as clinical pharmacokinetics or clinical pharmacodynamics. So where you are going to apply the PK and PD. PK/PD is pharmacokinetics, pharmacodynamic principles for the safe and effective therapeutic management of the drug in the patients. So we now try to touch upon these two concepts, pharmacokinetics and pharmacodynamics, a little more elaborately, because the entire discussion on drug interactions is based on these two aspects, pharmacokinetics and pharmacodynamics, because drug interactions can be via pharmacokinetics or via pharmacodynamics. So what is pharmacokinetics? Pharmacokinetics consists of four important processes. One is absorption, distribution, metabolism, and excretion. We call ADME. So absorption refers to the passage of the drug molecule across the barriers and reaching the circulation. And once it reaches the circulation, The drug is, generally it's in the free form. Free means it is not bound to anything. Many drugs are bound to plasma proteins and some are bound to the tissues. So this protein binding is a very important concept that needs to be considered. And this is part of the distribution process. So following absorption of the drug into the circulation, it gets distributed to different parts of the body. For example, it can go into the brain, it can go into the heart, it can go into every tissue, and then, but not identical rate. It can take its own rate, the entry rate into each of these organs or tissues. But eventually, as I said, since drug molecules are foreign molecules, the body doesn't want them. Therefore, it tries to get rid of them. And in order to facilitate its removal from the body, the systems within the body, like the liver, skin, and several organs, they have several metabolizing enzymes, and these metabolizing enzymes, they convert the chemical structure of the molecule into multiple metabolites. It is either one or many, depending on the nature of the chemical nature of the drug molecule. And once the metabolism takes place, the drug molecule is excreted via the portals of excretion. It can be bile, urine, saliva, sweat, and so many organs. So absorption, distribution, metabolism, and excretion, and that is what we call absorption, distribution, metabolism, excretion, ADME. And metabolism and excretion, these two can be combined. You call it absorption, distribution, and elimination. So ADE, that is, E there stands for elimination, which is the combination of metabolism and excretion. Or you can also remove distribution, and distribution, metabolism, and excretion together can be called as disposition. So absorption and disposition. That's good. So once the drug is absorbed, for example, say the curve that you are seeing here is following extravascular administration. Extravascular meaning it can be oral or it can be intramuscular, subcutaneous, wherever the drug has to cross the barriers and reach the circulation. And then when you start measuring the drug concentrations in the blood or plasma, the concentrations gradually increase and they reach a peak where the absorption is complete. And then what you see from then onwards is only elimination, which is consisting of metabolism and excretion. Although these two processes do happen right from zero time, but you don't see them because the drug concentrations are increasing because of absorption. So this particular typical curve pattern is, we call pharmacokinetic profile or PK profile of the drug. And corresponding to these drug levels, you also see the drug concentrations increasing at the site of action. And we call it as the target. We already defined that. And there could be other sites where the drug molecules go and bind, but may not result in any productive or fruitful drug response or efficacy. We call those as off-target interactions. So on-target interactions and off-target interactions. On-target interactions are the targets of our interest, and off-targets are targets which are not of our interest, and probably these off-target interactions can result in more adverse reactions. Okay, and apart from that, the higher the concentration of the drug, the peak, the effect that you are seeing is going to be influenced. So the higher the peak, the higher is the peak response. Likewise, the area under this plasma concentration time curve, we call it as AUC, is a pharmacokinetic parameter that will tell you the extent of availability or exposure, and that will give you how large is the exposure or how large is the effect. So how high is the effect and how large is the response. So these are the two that you could measure. And for every drug, you can define approximately two levels. One level is, something like a target level, and another one is a level below which the drug is not considered to be effective. That means in order for a drug to be effective, it has to cross a certain concentration threshold, and then only it shows its effect. And any level lower than that, you may not appreciate the pharmacological response or the desired response. And this is called a minimum effective level, this line. So this green area, what you are seeing between the two lines, we call it as the therapeutic window. And the drug levels above that, we call the adverse response. So that means the probability of drug producing more toxic effects or adverse effects is higher as the concentrations go beyond. It does not mean that in every individual you have got a fixed level and every individual will start having the adverse response above that particular level. It is only, some kind of imaginary area, imaginary line. So people may have start experiencing adverse effects right from here, and some people may not experience adverse effects even after reaching this point. So tentatively, you could say there is a therapeutic window, and you try to maintain the drug levels to the extent possible within this therapeutic window for optimal pharmacological response. And the broader the window, the broader is the margin of safety of the drug. The narrower the window, the narrower is the margin of safety. For example, if a drug is administered by IV route, intravenous route, directly into the vasculature, there is no need for absorption because the drug is already there. Within no time, the drug gets distributed in plasma and the concentrations will start going down. And if it is the same drug, you expect these two, the elimination processes to be parallel to one another. And generally, we define pharmacokinetics using certain pharmacokinetic parameters. Many of you must have read about these, like clearance, volume of distribution, absorption, half-life, area under the curve, about which I already mentioned, Cmax, that's the peak concentration, and Tmax, which corresponds to the Cmax, the time at which the peak concentration occurs. And clearance is a very important parameter because this tells you about the true elimination behavior of the drug. That means how quickly the drug is removed from the body. So there are certain situations where the drug can be more rapidly cleared, and there are situations where drug is less rapidly cleared or slowed down. And this is one parameter which is likely to be affected by another drug. So a drug may interact with the first drug by affecting its clearance or by affecting volume of distribution, and thereby it can cause alterations in its pharmacokinetics. And once pharmacokinetics is altered, the extent of exposure, the peak exposure, the Tmax, the time at which peak occurs, and the half-life of the drug, any one of them or multiples of them can be affected, and that can result in an interaction. So this can be related to pharmacodynamics. I already mentioned pharmacodynamics is the science where you are trying to quantify the drug effects, and there is a good relationship between the drug concentrations in the plasma and the drug effect. For example, heart rate reduction by an antihypertensive drug. So the concentration of the antihypertensive drug, if you put it on x-axis and y-axis heart rate, that the percentage reduction, is changing probably quite linearly. So you can't expect this kind of linear relationship between drug concentrations, that is pharmacokinetics, and the drug effect on y-axis. This is linear, this is great, but many a time it is quite possible that the effect is not likely to be linear. You will find some kind of curve pattern like this, we call sigmoid curve. Initially it is slow, and then it becomes linear, and then again it becomes a plateau, and you will find the curvilinear space. So only thing that you need to take into consideration is if you want to express this mathematically, you may have to use a bunch of equations. I'm not going to touch upon those. It is beyond the scope of today's discussion. And the curves can also show various slopes. And this again depends upon the particular molecule under consideration. So now we can link these two, pharmacokinetics and pharmacodynamics. Just now I mentioned in the plasma or in the blood, the drug concentrations increase, reach a peak, and then fall. And the drug has to cross the barriers in order to reach the target site. For example, somebody has a problem in the brain. Say, for example, patient has got epilepsy, and so the drug has to get into the brain and has to do something there. And in order to do something there, it has to bind with the receptors or some kind of target molecule. So this is the concentration of the drug in the site, at the site where the drug produces its effect. And at the site, once it starts binding, there is an interaction between the drug molecule here and the drug target, that is a receptor. And there is a dynamic equilibrium between the two. And you can actually measure the effect versus time. So this is pure pharmacodynamics. This is pure pharmacokinetics. These can be. Okay, so this is very important basic introduction I thought I should provide. And if all of you, or if many of you already know about it, I apologize for taking or wasting a few minutes time. Now we will move on to drug-drug interactions. So why do drug-drug interactions matter at all? And since the topic of my today's discussion is to touch upon the clinical and regulatory aspects, so it is very important from both sides. So clinical drug-drug interactions are a leading cause of preventable adverse drug reactions. So this has been already envisaged by Dr. Kalai that ADRs, most of the ADRs today are caused because of drug-drug interactions, particularly in the advent of polypharmacy. That means people using multiple drugs for various indications simultaneously. And second, from the regulatory perspective, drug industries are involved in developing more and more new molecules, new drugs, and they are brought into the market. And you know pretty well that a new drug takes about 10 to 15 years of time and several billions of expenditure. And you do not know about the drug's safety, particularly with regards to its drug interactions, until recently, until the drug has been approved and launched. So people have been using it in the clinic. And some doctors, they come back saying, oh, this drug is causing this problem. And so immediately, the drug is kept under, big microscope, and the regulatory authorities will start working on, the problems associated with the drug molecule. And like that, several drugs have been scrutinized in the past based on very severe or serious adverse drug reactions. And that has led to the even withdrawal of these, unfortunately, not without much of damage being done. Several people died because of unanticipated adverse drug reactions. One of the classical examples is terfenadine. Many of you are aware that it is one of the, when it was discovered, it was considered to be one of great drugs because, it is a non-sedating antihistamine. And this terfenadine is a prodrug, incidentally. And inside the body, it is converted by the action of a particular metabolizing enzyme. And then it is converted to active molecule. Therefore, it is a prodrug. But in some individuals, it was found because of a drug interaction, terfenadine is not getting metabolized and it is remaining as it is. And if it remains as it is, it causes very important kind of arrhythmias. And this is because of the QT wave prolongation in the ECG. Likewise, another drug, a statin drug like atorvastatin, rosuvastatin, simvastatin, so many statins have been there. And the serivastatin is one of those. And this particular drug also, due to drug-drug interactions, it develops what is called as rhabdomyolysis. That is nothing but the degradation of the muscle fibers, resulting in very severe toxic side effects. And as a result, even this drug has been removed from the market. So is there anything that the regulatory agencies could do to prevent this from happening? But that too, after spending so many billions of dollars and spending so much of time on the drug being approved by the agency as a new drug, and then launching and then perceiving its potential to cause enormous toxicity. So some of these aspects are nuances we are going to cover in today's talk. And generally in the drug-drug interactions language, we use two specific terms. One of them is called perpetrator or called precipitant, the one which causes drug interaction. The other one is victim. This is what is called object. So perpetrator and victim. Drug interaction is caused by so-and-so drug, and drug interaction occurs to some such and such drug. So these are the two we always talk about. So perpetrator is the drug that causes the effect, and the victim is the one which is victimized. For example, in the case of terfenadine example, terfenadine is the victim. Surfastatin is the victim. So, okay, what are the perpetrators there? We will see later. And then the scope is, as I said earlier, it can be drug food, drug herbal extracts, interactions. So once again, I told that the drug interactions can be evaluated or studied from the perspective of pharmacokinetics as well as pharmacodynamics. So if the pharmacokinetics are altered, then it can cause drug interactions. If the pharmacodynamics of the drug are altered, then also it changes the drug effect. So either way, it is possible drug interactions can occur, but most predominantly we see pharmacokinetic interactions causing a lot of problems in the clinical practice. So pharmacodynamics, it's pretty clear. The drug concentrations are not much changed. They're pretty much the same by the so-called, the predator drug or the precipitant drug. But in the presence of one drug, the other drug's activity, the toxicity is increased, or it will have an opposite effect. So we will see some examples moving on. Like for example, if somebody is taking an opioid drug like oxycodone, if he also consumes a benzodiazepine, it can cause a severe respiratory depression. So this is an additive effect or a synergistic effect. We call additive if the combination is the sum total, and it's called synergistic if the net effect is more than the total, sum total. And antagonistic effect is opposing effects, like a beta blocker, non-specific beta blocker, which blocks both beta 1 and beta 2 receptors. Beta 1 are located in the heart and beta 2 are located in the bronchial smooth muscle. So a drug like propranolol can cause an opposing effect when somebody is taking a beta agonist as a bronchodilator. So there is an opposing pharmacological effect. So this is called the antagonistic effect. So we will see more examples. And as I mentioned, that pharmacokinetics consists of 4 important processes: absorption, distribution, metabolism, and excretion. We will touch upon each one with a few examples. So absorption is gatekeeper. Absorption is taking place from the gastrointestinal tract. Absorption is taking place from the muscle, from the skin, under the skin, and so on. So this is the gatekeeper interaction. So absorption interactions can occur when you take a tablet or when you take a capsule or a suspension orally because of chelation or complexation or pH alterations or adsorption, which is a surface phenomenon, or GI motility changes. That means one drug may complex another, or one drug may alter the pH of the gastric environment, or one drug or one substance may adsorb the active ingredient, and one drug may alter the gastrointestinal motility, thereby it affects its pharmacological effect. So the first example is chelation, is very, very common example. Many of us know that the calcium, magnesium, iron, aluminum, these are components of several drugs we use, for example, iron supplements, antacids, and several multivitamins consisting of so many minerals in it, and likewise dairy products which contain proteins, calcium, and so on. So these, they bind with the drugs, bind to electron-rich drugs, and thereby they form complexes, and these complexes do not get absorbed. Once they do not get absorbed, they cannot produce the pharmacological response. So the perpetrators here are antacids, iron supplements, and so on. The victims are very classical example, tetracyclines like doxycycline, oxytetracycline, etc. Several fluoroquinolones, example ciprofloxacin. If you take them along with any of these antacids, etc., you can expect almost 50% to 90% reduction in bioavailability. So what happens if bioavailability is cut down by 50 to 90? Obviously, the patient doesn't respond to the antibiotic. So what you can do, the remedy is pretty simple here. You space the dose. Don't take these two together because the interaction is pretty immediate. So let there be at least 2 hours before or 4 hours after administration of these antacids or the perpetrators, you can take the drug. So that is one, complexation. Number 2 is acid trap. That means if you alter the pH of the acidic environment of the stomach, For example, several drugs that we use are weak bases. For example, antifungal drugs like itraconazole, ketoconazole, and then the tyrosine kinase inhibitors, the dasatinib. I just included only 3 examples here, but there are multiple drugs are weak bases. And you know pretty well, weak bases are readily soluble in the gastric acidic environment. And if you administer any drug that elevates the gastric pH or raises the gastric pH, like for example, omeprazole is an anti-ulcer drug, protein pump inhibitor, or H2 receptor antagonists like famotidine, ranitidine, etc., or simple antacids, they can neutralize the acids and elevate the pH. And once the pH crosses 4, these weak basic drugs, they will not get ionized easily, therefore they will not get dissolved. And once they don't readily get into solution, their absorption is impaired. And so there is the delay in the absorption, or there could be a decrease in the absorption. So both things can happen. So how do you manage? Very simple. You don't use that drug. You use a drug that is whose solubility is not dependent on pH. Instead of choosing, say, itraconazole, the physician can think of using fluconazole for a patient who is on, proton pump inhibitor dosing. Or you can play a small trick, that is, take something that acidifies your stomach environment, like take a glass of Coca-Cola. But thing is, you can't advise the patients to take Coca-Cola every time he consumes the drug. And the next absorption kind of interaction is the absorption and motility. The absorption, I told, is nothing but a surface phenomenon. There is a complexation happening. For example, ion exchange resins like cholestyramine, which sequesters bile acids, cholesterol. In fact, it was used earlier before the introduction of the statins for binding cholesterol. So it is used for binding or reducing the absorption of cholesterol and many toxic substances. So is the case with the activated charcoal. It is used in several poisonings, particularly alkaloidal poisonings, because it absorbs these and thereby prevents the absorption into the system. So generally, the victims are warfarin, digoxin, and several other molecules which are administered concomitantly whenever cholestyramine is used. So once again, you provide a gap between administration of this perpetrator and the active ingredient. And then the motility influencing agents like prokinetic agents, metoclopramide, you know pretty well that there are several hundred combination drugs available on Indian pharmaceutical market consisting of metoclopramide and acetaminophen, that is paracetamol here. So what is the meaning of this particular combination? Metoclopramide, it hastens the speed of motility of the gastrointestinal tract. So thereby it facilitates rapid gastric emptying, so that acetaminophen gets into the duodenum, small intestine. So where its absorption is pretty rapid. So you are enhancing the onset of action of acetaminophen by putting it along with metoclopramide. But in several other countries, they consider this particular combination questionable, because why do you administer metoclopramide, which has its own pharmacology, along with acetaminophen, unless it is pretty much required. Likewise, anytime you administer an opioid or an anticholinergic drug, it slows down the gastrointestinal motility. Therefore, any drug like digoxin, its absorption is delayed because of the slowing down of the intestinal motility. So I think these are very fundamental, basic concepts. Every practicing physician, I believe there are some doctors on this call. So my suggestion to them is that every time you are dealing with drugs which have a potential to alter the gastrointestinal motility, please be mindful whether you are dealing with a drug which is potent and whose the alteration of absorption can result in drastic changes in its pharmacology. So then coming to the protein binding, which is part of distribution, I told distribution is nothing but, once the drug enters the circulation, it goes to different tissues, different parts of the body. And the most important place where the drug gets bound is the plasma proteins itself. In plasma, there are 2 kinds of proteins, which is albumin, and the other one is alpha-1 acid glycoprotein. We call it as AAG, alpha-1 acid glycoprotein. This is albumin strongly binds the acidic drugs, and AAG strongly binds the basic drugs. So pretty much you know about the binding characteristics of the drug. Therefore, you would know how much it is normally bound. You should also have some basic information about the perpetrator drug, whether it is capable of displacing the victim drugs binding to the plasma proteins. Because some drugs are bound to the extent of almost like 99 or 99.9% even. So in such instances, administration of a perpetrator, which also has the binding affinity to the same sites, because every protein has got specific binding sites, and these binding sites, they are probably unique for that particular molecule, but any other drug administered concomitantly can bind to the same sites and can displace it. So displacer and victim. So there are many examples, and we are going to talk about a couple of examples later. And so what happens when a drug is removed from the binding sites in plasma? There is going to be more free drug available. You know very well, the drug that is bound is not active. The drug has to be in free form in order to be active. So if you have a drug that is 99% bound to the plasma, so that 1% of the drug that is in the free form or unbound form is active, whereas the remaining 99% is inactive. So when you add a drug that displaces it from the active binding sites, it is going to cause or liberate more free drug. So at the outset, it looks as though it is going to cause a serious problem. Probably it is not. We will see the reasons later. So then comes a very important class of interactions. These are pharmacokinetic interactions, where the interactions occur at metabolism and transporter sites. So coming to the metabolism is nothing but chemical conversion. So why would metabolism take place at all? The purpose of metabolism is to convert a drug into more easily excretable form. This is what generally happens, or more water-soluble form. At times, it doesn't happen. Drugs can become more water-insoluble, and many a time it happens that water solubility of the drug molecules increases. And it is also possible that the metabolism can be a single phase or it can be a double phase. Single phase can be phase 1 or phase 2, and at times it can be phase 1 plus phase 2. Phase 1 reactions are called as the functionalization reactions. I don't want to go too basic here. And the most important enzymes responsible for phase 1 metabolism, these are called cytochrome P450 system. This is called CYP system, or cytochrome P450 is a super family of enzymes. It consists of several hundreds of enzymes, and it is a super family consisting of several families, and these families consist of several isoenzymes. And nearly 80% of the drugs that we are using today are metabolized by cytochrome P450 enzymes. So now what happens in a drug-drug interaction situation? So you are having a perpetrator and a victim. A victim is a drug whose metabolism is altered. Perpetrator is a drug which is responsible for its disalteration. So it can be inhibition or it can be induction. Inhibition can be competitive. That means the two drugs are, when they are there simultaneously, competing for same cytochrome P450 isoenzyme. So one of them wins, the other one loses. The loser is the victim, and its effect is compromised. And at times the inhibition can be reversible, because something basic, more permanent destruction of the enzyme happens. As a result, the victim is victimized forever. And the second one is induction. That means administration of a perpetrator, that is the interacting drug in the presence of a drug that is a victim. This does not happen immediately like inhibition. It takes time, probably a week or even longer than a week. So you keep on administering the perpetrator drug, and the metabolism of the first drug gradually keeps on increasing. It becomes more and more and more rapid. And after some time, its levels become so small that its activity is lost. So this is what is called the induction. Inhibition and induction, inhibition is generally immediate threat, whereas induction is a delayed threat. So this is just to give you an idea, like how the superfamily looks like. Cytochrome P450 monooxygenase enzymes is also called as mixed function oxidase system, where the enzymes are not specific for a particular drug or particular structure, but these are more mixed function. That is, several drugs can use a particular isoenzyme for its metabolism. For example, 30% of the drugs are metabolized by CYP3A4, that is shown here in blue form. And likewise, the CYP2D6 is another very important isoenzyme. CYP2C9 is another, CYP2C19, CYP1A2, and CYP2A6. These are very commonly participating enzymes. And these enzymes, they express, several, forms within themselves. That means an individual may not express the same amount of enzyme, and this is genetically influenced. And some people may express a good amount of enzyme, and some people may not, express the same amount of enzyme. So this is what is called as the polymorphism. That means people expressing different levels of enzyme because of certain genetic mutations, and these mutations result in what is called as the polymorphism. For example, if you take CYP2D6, it can be divided into multiple forms. Some individuals, when you administer a particular drug, they metabolize the drug extremely rapidly. We call them ultra rapid metabolizers. Some they metabolize rapidly, some they metabolize slowly, some they metabolize intermediate, and some metabolize it very, very slowly. So depending on what kind of subgroup you are falling in, the way you are handling a particular drug is going to alter, and as a result, its activity is also changed. So there are so many genetic variations, and this is the subject of discussion under what we call a broad area. Today we call it pharmacogenomics, and that is leading to the concept of individualization of the drug dosing or drug administration. So we'll take a bunch of examples here and try to focus. You don't have to remember the names of all the drugs. I just took a couple of studies here. A 30-year-old guy, he has backache, severe spasm, and the doctor has prescribed him a drug called tizanidine. This is a centrally acting skeletal muscle relaxant. And this particular molecule upregulates alpha-2 adrenergic activity centrally. Alpha-2 receptors are the adrenergic receptors. So these are present abundantly in the brain. So tizanidine causes upregulation of this activity in the brain. And the individual also happens to have a UTI, urinary tract infection. Therefore, the physician has prescribed ciprofloxacin 500 milligrams twice daily. And after very first dose of the antibiotic, the individual who has been on tizanidine treatment, an hour later, he is found unconscious. With when the BP was measured, it was 70/40. So the mechanism here is that as I told earlier, the inhibition of these cytochrome P450 enzymes is can be very, very rapid, sometimes almost instantaneous. So here, inhibition of the metabolism of tizanidine is taking place because tizanidine is the substrate for CYP1A2 enzyme. So once it is inhibited, the drug levels in the brain for tizanidine are increased. So alpha-2 adrenergic activity is tremendously increased. So as I mentioned earlier, AUC, area under the curve, this is the systemic exposure of tizanidine is increased by 7 to 10-fold by a single tablet of ciprofloxacin. That has resulted in a massive vasodilation and sedation. So there is no surprise that the patient has become unconscious. So what is the remedy for this? You don't use these two together. Or better thing is, you take a drug, an antibiotic that can be can be used in place of ciprofloxacin. For example, levofloxacin does not inhibit CYP1A2. So replace ciprofloxacin with levofloxacin. Simple. And then another kind of situation can also happen. This is CYP2D6, where we are talking about a situation where a female patient having a very, mild to moderate back pain has been on codeine. So codeine is being used in certain parts of the world, particularly in the western part of the world. In the US, codeine, not always, but except in certain individuals, codeine can be administered and it is being used. And codeine as a painkiller is very mild and moderate in its activity. As such, it doesn't produce significant pain relief, but it is converted into morphine. Morphine is the pain reducing molecule, which is a metabolite of codeine. And this metabolic conversion is carried out by the enzyme CYP2D6. So the patient has been taking codeine on doctor's advice, and the patient also has depression. And so she has been on, selective serotonin receptor, serotonin release inhibitor, that is fluoxetine. And after some time, she comes back to the doctor and saying that, she's not getting any relief from the back pain. So codeine is useless. So the doctor increased the dose, no chance. The problem still persists. And then when it was further investigated, it was found that fluoxetine is a very powerful inhibitor of CYP2D6. Simple. So the other perpetrators are paroxetine, buprenorphine, quinidine. So these are CYP2D6 inhibitors. And where do you get this information? This information is all over. So whenever somebody is put on codeine, the physician is expected to be aware that codeine is not the drug that produces pain relief, but morphine. And that particular drug, has to be converted into morphine. And drugs like paroxetine and other antidepressants and non-antidepressants like quinidine, for example, an antiarrhythmic drug, can inhibit this particular enzyme, and thereby it blocks conversion into morphine. So this is very, very important interaction. I came across one interaction related to CYP2D6. So I give a specific significance to this particular enzyme because we very commonly come across situations where CYP2D6 interactions occur commonly. It could happen in case of lactating mothers. There was a case study reported in Lancet where a mother lactating a baby, very healthy baby, days-old baby. She was receiving codeine for pain after the childbirth, and then after some time the baby developed respiratory depression, and that's because of excessive conversion of codeine. The reason there was that the woman was carrying 2 alleles of the gene that expresses CYP2D6 enzyme. And as a result, the mother has been very rapidly under large amounts of codeine into morphine. So the baby died because of respiratory depression, as morphine was, received via the breast milk. So whenever you are dealing with this kind of interactions, we have got to be extremely careful. So this is about, I can give you hundreds of examples related to cytochrome P450 enzymes, but because of the paucity of time, I'm skipping. Just to put it in perspective, a drug that is administered, it may or may not undergo metabolism. Some drugs may not undergo metabolism at all. So they may be excreted directly via urine, that is through kidneys, or may be eliminated via liver, that is via biliary tract as bile through the feces. But otherwise, those drugs which undergo metabolism, they undergo either phase 1 metabolism mediated by oxidating, reducing or hydrolyzing enzymes. And after conversion into a metabolite, it undergoes further conversion to form conjugates. So this is called phase 2 metabolism, and these are called conjugation reactions. Many of you, if you could just refresh your memory, I'm sure you must have read these in your biopharmaceutics or medicinal chemistry, wherever. And once it is converted to phase 2 metabolite, these are conjugation metabolites. These are pretty big in size, molecule size, and they are more polar generally, and they are more easily excreted. And many of them are inactive. There are exceptions, and one of the exceptions is morphine glucuronide. Morphine 6-glucuronide is a more potent analgesic than morphine itself. So these are the phase 2 metabolizing pathways. I'm not going to explain all these, but just remember, the group that is added to the molecule is glucuronic acid, or it is a sulfate group. If you have phenol or steroids, the sulfation occurs. If the drug molecule or the metabolite has an amine group like isoniazid and dapsone and drugs like where there is an amine group freely present, then N-acetyl transfer occurs, they become acetylated, and that is another. Or methylation can occur, like for example 6-mercaptopurine or azathioprine. Some of these drugs, they are conjugated, thereby they produce corresponding methyl derivative. Couple of examples related to phase 2 drug interactions is that one very interesting example is in case of epileptic seizures, a patient has been receiving lamotrigine. Lamotrigine has got two effects. One is it helps in reducing seizures, and it also helps in stabilizing the mood. That is, it can be used in bipolar depression. And the patient also required valproic acid according to the physician. Physician thought the patient requires another anti-epileptic drug to be added, but the physician has not considered the potential interaction between valproate and lamotrigine. Valproic acid, is a very potent inhibitor of this particular UGT1A4, which is responsible for the glucuronidation of lamotrigine. And what happens? Lamotrigine metabolism stops because it doesn't get UGT1A4, and as a result, the lamotrigine levels will increase 2 to 3 times, and thereby it drastically increases the adverse effects. And the most important adverse reaction that is commonly seen with lamotrigine is development of very extreme, severe, sometimes life-threatening skin reactions called Stevens-Johnson syndrome or toxic epidermal necrolysis. I think many of you are aware of this particular reaction. And how to mitigate this reaction? Simple. What is happening here is the drug levels of lamotrigine are increasing. Therefore, the physician should preemptively means that, the beginning itself, before adding valproate, should cut down the dose of lamotrigine by 50% or even more. And another very interesting example, this is another classical example where, with the oral contraceptives, and this may not happen all the time, but there were reports where oral contraceptives, when they are administered, antibiotics could alter the oral contraceptive levels, and thereby they can cause a failure of the oral contraception. That oral contraceptives, these are steroid nucleus, that is progestins, etc. When they are administered, they are converted into their corresponding glucuronides, and these are larger molecules. Therefore, they are released through bile, and they get into the intestines. And once they get into the intestines, what happens generally is that the intestinal bacteria produce glucuronidases, that is enzymes that cause the conversion of glucuronidated metabolite of oral contraceptive molecule, and liberate the oral contraceptive in free form, and the free form is reabsorbed. So this is what we call as the enterohepatic recirculation. So drug gets into the circulation upon administration, gets metabolized, converted to glucuronide. Glucuronide is eliminated through bile, gets into the intestines, and intestinal flora. The bacteria will split it down into oral contraceptive again, and then that oral contraceptive is absorbed back. So if you kill the bacteria by using a broad spectrum antibiotic like doxycycline. Doxycycline is not that bad. Other tetracyclines or other broad spectrum antibiotics, then the bacteria are killed. Therefore, they will not be able to release oral contraceptive again, so that it is reabsorbed and produces the desired pharmacological effect. So there is a possibility of therapeutic failure, and so once there is therapeutic failure of oral contraceptive, there is an unplanned pregnancy. And then comes another very important concept, another site where drug interactions can occur. This is a little more intricate. Give me just a half a minute. Yeah, we call these are transporters. So transporters are very important systems which you have them at multiple sites in the body. You have them in the intestines, we call enterocytes, the intestinal cells. The drugs get absorbed from the intestine, cross the enterocyte, and they get into the bloodstream. Likewise, you have hepatocytes, that is the liver, where the drug molecules from the circulation enter into the liver, and then something happens within the liver. Likewise, in the kidneys, particularly in the proximal tubules, there is transportation across the barrier, the intestinal proximal tubular epithelia, and likewise even within the brain, blood-brain barrier, the blood-brain barrier endothelial cell lining, there too you have got certain transporters. And the placenta also you have transporters. So although they look, many, it's very simple if you can focus just a little bit. And this is a very important concept. I think those who have been, doing good amount of work in the area of transporters, this will be helpful. Not only that, this is one area where the regulatory has come out with certain new guidelines based on the International Council of Harmonization guidance, M12, where how to perform drug interaction evaluation in the drug development. So these are, there are a couple of them which are responsible for the efflux of the drug molecules, and there are a couple of them who are responsible for the influx into the system. So these are organic cation transporter, for example, here, for the drug to enter into the system, and the P glycoprotein, and then breast cancer related protein. These are the efflux proteins. They throw the drug out from the enterocyte into the intestine. So the drug that is interacting can inhibit the drug which is the victim that is taken up by the P glycoprotein, or OATP1. This is a transporter that you have. This particular transporter is responsible for pushing the drug molecule from the circulation into the liver for metabolism to occur. So I'm not going to discuss too elaborately about these. There are two types of transporters here. We call them as the ATP binding cassette. We call them ABC. Efflux transporters, and SLC influx transporters. That means some transporters take the drug molecules into the cell, and some transporters take the drug molecules outside the cell. So, and interactions can occur here. These are very, very important interactions. These are as important as the drug metabolism related interactions. So, for example, digoxin is a P glycoprotein substrate. And this P glycoprotein that is present in the intestinal lining, will not allow the digoxin to get accumulated, and this is going to throw it out when you administer digoxin. And in case if you are using a drug that inhibits this particular transporter, like amiodarone or verapamil or clarithromycin, what it does is it does not inhibit. Therefore, digoxin concentrations will be increased because of increased absorption. So this is an example which clearly explains this. So this is the intestinal lining. This is P glycoprotein present here, a transporter. Digoxin is a substrate. Usually digoxin is not allowed. It is transported by PGP and thrown out into the intestinal lumen. But if you administer amiodarone enters into this enterocyte. It inhibits this transporter. And once this is inhibited, digoxin is not stopped to be removed outside, and so it remains here, and its circulation, its concentrations in the circulation will increase, and it produces its toxicity. So likewise, another example, this is about hepatic transporters. So this transporter is called organic anion transporting polypeptide. These are again very important gateways. What they do is they allow the entry of drugs. Classical example is statins. All the statins that we take, they pass through these transporters, get into the liver, And there, unless the statins reach the liver, they cannot produce the anti-cholesterol effect. Okay, they cannot reduce the elevated levels of cholesterol. And so if you administer a drug, a drug like rifampicin, or there are other drugs, cyclosporine or gemfibrozil or ritonavir. So these drugs, they cause inhibition of this particular transporter, OATP1B or B3, and so the drug cannot get in. And once it cannot get in, it's not producing its hypercholesterolemic effect. That is one thing, and it also cannot get metabolized. Therefore, the drugs levels in the plasma or blood are increased tremendously, and once it happens, it will produce a lot of its side effects. Like, for example, rhabdomyolysis, as I mentioned earlier, can cause muscle breakdown, severe muscle cramps, pain, nausea, and so many other things. And likewise, interactions can occur within the renal tubule, the proximal tubule, where there are two kinds of transporters. One kind of transporter is transporting from the blood into the proximal tubule, and another transporter that is responsible for throwing the drug from the tubule into the urine. A drug like cimetidine, can inhibit OC2, this transporter. And what happens if metformin is the substrate, and metformin removal is impaired once this is inhibited. So its accumulation happens in the circulation. So once metformin levels increase in the blood, what happens is metformin the effects are increased by several fold. It can cause lactic acidosis, and it can also cause a severe hypoglycemia and so on. And it's not only that, cimetidine also inhibits this transporter called MATE transporter, called multi-drug and toxin extrusion transporter. And this will result in accumulation of metformin here. Once it is increased here, it will get back into the blood. So we have got to be very careful about this. And then coming to the pharmacodynamic interactions, I already mentioned earlier, the concentration is not affected, but pharmacology is changed. Additive is, if you add 1 and 1, it is 2. Synergy is, if you add 2 drugs, the pharmacological response is more than 2, and it can be beneficial or toxic. And the antagonistic is, as I mentioned earlier, a non-specific beta blocker plus a beta 2 agonist. So these are very classical examples. Many of you are aware. I just quickly touch upon these. This particular combination is being used in regular therapy, and you have marketed products like Augmentin, which has amoxicillin and clavulanic acid. So this clavulanic acid, it inhibits the beta lactamase producing bacteria. Therefore, beta lactamase, it selectively cleaves amoxicillin and makes it inactive. And so presence of clavulanic acid, although it doesn't contribute much to its antibiotic spectrum, it protects amoxicillin, thereby it enhances its pharmacological effect. So I don't want to dwell much into this. I think many of you know this classical example. This is a very classical example, and I want to take a minute here. We were talking about terfenadine. So what terfenadine does is, it is rapidly converted into its active metabolite, which is called fexofenadine. So if you inhibit the metabolism of terfenadine, it is not going to be converted into fexofenadine, and it can cause inhibition of the potassium channels. These potassium channels are called as HERG channels, human either a go related gene potassium channel. Try not to memorize it. It's a very important potassium channel, and some drugs have got the propensity of inhibiting this channel. And once it is inhibited, it causes prolongation of this QT, wave or interval, and this can be very clearly demonstrated in an ECG. So you could see here, this is uninhibited QT, and the QT interval is so much. And if it is prolonged, or if a drug inhibits this channel, HERG channel, there is so much of prolongation. So normal male subjects should have the QTC interval, less than 440 milliseconds, and women less than 460 milliseconds. And if the QTC, this corrected QT interval, if it is more than 500 milliseconds, the patient will suffer from a typical kind of fatal arrhythmias. It is called as Caussady point A. So this is generally seen whenever there is an interaction between one drug and the other. For example, a drug called sotalol, many of you know about it. It's a class 3 antiarrhythmic. And if you administer levofloxacin simultaneously, this happens. It inhibits the metabolism of this drug, and it causes, prolongation of the levels increase. And as a result, it can cause prolongation of QT interval. Same thing with terfenadine. So there are several examples in the literature, and so the regulators are very, very concerned, and it is one of the very important, important channels or toxicity related flags that the agency always looks for in the early development of a drug molecule. So it is possible to assess it in vitro interaction. And another classical PD interaction is, one drug blocks the other. For example, this is a very common scene in many countries where, opioid abuse takes place and people consume overdosing of, people consume opioids. And overconsumption can result in, the important the agonistic activity of the mu receptors present in the central nervous system. And, this can cause a respiratory depression and death. So when somebody is getting into respiratory depression, today we have Narcan nasal spray. You can just simply use this naloxone, which antagonizes the action of opioids and produces immediate clinical response. This is a classical and well-known example. Likewise, a bleeding condition, people who have been on aspirin treatment, if they also happen to take warfarin, an orally effective anticoagulant, which decreases production of several blood clotting factors like 7, 9, 10 prothrombin, there is excessive, anticoagulant effect that is produced. Not only that, aspirin causes gastric insult, and it also causes platelet deaggregation or inhibits platelet aggregation. And at times, people may use ibuprofen either in combination with aspirin or combination of both warfarin and aspirin. This adds to the mucosal insult or injury even more, can cause a massive, bloody situation resulting in sometimes death. So you have to be very careful whenever you are dealing with, drug coagulation affecting drugs or antiplatelet drugs. And another example where drug can influence its reabsorption is in the renal tubules. Another classical example, aspirin when taken, it is reabsorbed when it is in acidic form in the renal tubules because it is in the unionized condition. And if there is aspirin poisoning or salicylic acid poisoning, people generally administer sodium bicarbonate. Thereby, you are going to alkalize the urine, and in presence of an alkali, the acidic drug like aspirin is converted into its ionized form, and ionized form is not reabsorbed, so it is eliminated. So you are making use of an interaction for a beneficial effect. So now, we will touch upon some of important points in the end towards the latter part of my discussion. So identifying high risk scenarios. This is the message for the people who are in the clinic, either doctors or clinical pharmacists with the PharmD qualification, or those who are dealing with the clinical aspects related to the drug-drug interactions. Whenever you are dealing with drugs which have been labeled as narrow therapeutic drugs, or I already mentioned what narrow therapeutic index is, narrow margin of safety. So these drugs, have got to be watched with a lot of care. Like for example, warfarin, digoxin, cyclosporin, tacrolimus. These are drugs. Whenever you are dealing with drugs like these, you have got to be immediately. The light should, brighten up in your mind. Okay, we need to be careful. These are red flags. And then whenever we are dealing with a vulnerable population like fragile individuals or people who are having protein malnutrition and other conditions like severe edema, obesity, elderly and people have, above 65 even, they are possibly receiving multiple drugs, we call polypharmacy, and they will have generally some kind of reduced renal function, compromised renal function, and that has to be taken into consideration. The reduced hepatic or renal impairment, alcohol consumption, which has become nowadays a very common feature. For example, the dose of acetaminophen, FDA has very stringent instructions related to the paracetamol dosing. Although it is indicated for normal healthy individuals, it is 4 grams. It has to be cut per day. It has to be cut down to even 2 grams, depending on the alcohol drinking status of the individual. And above that, I already talked about pharmacogenomic implications. We discussed codeine toxicity in a newly born baby. This can happen even in adults. So you always have to have this in back of your mind, the pharmacogenomics of the metabolizing enzymes. So the clinical management requires 3 strategies. One is you always monitor wherever it is possible, particularly for drugs which have narrow therapeutic index, like warfarin or digoxin, and anti-epileptic drugs. We have what is called therapeutic drug monitoring, and then adjust the doses preemptively. This also we talked about spacing the dosing of the perpetrator and the victim, and minimizing. That means don't unnecessarily prescribe. If you are pharmacist working in a hospital where the physician has a tendency to prescribing several drugs in the prescription, like 10, 15, some of them are useless. They think they are innocuous or they're harmless, but they can cause life-threatening drug interactions. So from the regulatory perspective, my suggestions to the regulatory guys are the regulatory divisions all across the world now, particularly FDA, EMA, and other regulatory agents, have been taking more proactive kind of role. Thanks to the development of clinical pharmacology last 30 years, very recently, this particular, the ICH guideline, ICH M12, that is related to the drug-drug interactions, has been promulgated 2024 May, if I'm not wrong, was issued. And this gives you very clear instructions as to how the drug discoverers or the pharmaceutical industry should tackle this issue right from the very early stage of drug development. So the old paradigm was like, the drugs were introduced into the market. So several phases have been passed through. Phase 1, phase 2, phase 3, a drug is launched, and then physicians start prescribing. And it is possible that the patients may come back with the drug interactions, and perhaps at that point of time it is already too late. And so it is necessary to have these, instead of handling these drug interactions as something like a reactive strategy, it has to be more proactive strategy. That is what the new paradigm. For example, FDA now has a very stringent requirement that all the drug developing companies should start working related to drug-drug interactions right from the beginning. So a risk-based approach to determine if a clinical study is needed. So you will have to consider that. And there are certain models that are prescribed in this M12. I'm not going into the details. You can assess the possibility of drug interaction in the individuals. For example, whether it is intestinal enzyme like C304 or systemic enzymes, you can assess the risk of interaction based on the inhibitory constant, and also what is the free drug concentration in the plasma. So this is the ICH M12 risk-based tree. I'm very sorry that the print is very small, but typically it is like this. You need to conduct in vitro screening, where you have to see whether there is any potential for interaction at the cytochrome P450 level in microsomes or cell lines. And likewise in the transporters, we talked about several transporters in the liver, kidney, and also intestines, etc. These are to be initially worked into. And if there is any early signal, if there is no signal, it is all right. But if there is a signal, you assess how risky it is. And if you think it could be potentially risky, then you will have to perform what we called some modeling. Today we are going to make use of what is called a physiologic-based PK modeling, or if the agency requires that, you may also have to conduct a limited number of clinical drug-drug interaction studies, generally in healthy subjects, so that you will have a clear idea whether whatever interaction that you have seen in vitro is significant in the human population. Just a word about physiology-based PK modeling, or PBPK, where you are going to consider physiology and the biochemical properties or physicochemical properties of the drug molecule. That is how the drug, upon entering into the bloodstream, gets distributed to different tissues. All this can be modeled. We have excellent software, like for example SimSip, GastroPlus, and we have what is called the PKSim, etc. These can be used for determining very effectively under different therapeutic conditions in animals, in humans, and so on, to predict the potential of drug interactions. And likewise, this PBPK modeling is also used for determining the disposition in pediatric population. So just a couple of case studies. You try to guess. I will give just less than half minute before I disclose the answer. Try to see whether you can come out with any answer to it. So case 1 scenario: patient on stable warfarin and oral anticoagulant therapy is prescribed amiodarone, is an antiarrhythmic drug. Guess what happens, why, and how to mitigate? Okay, since you see the name warfarin, you can pretty much at least come out with a guess that something has got to do with warfarin or warfarin activity, anticoagulant activity. Probably there is a delayed coagulant effect, and there can be a potential bleeding. If you can guess that much, I think the purpose of my talking all the earlier slides has been achieved. So there is one parameter we call INR core. It is known as International Normalized Ratio. This is just to measure how effective your warfarin treatment is in reducing the clotting. So if you administer amiodarone is the perpetrator. It inhibits this enzyme called CYP2C9. So what happens? The warfarin levels will increase enormously. So the maximum effect is seen in about a couple of weeks. So what is the remedy for this? So preemptive reduction in the dose of warfarin by 50%, because warfarin levels are increased in the presence of amiodarone. So in case if you cannot stop giving amiodarone, cut down the warfarin dose. Okay. And the second case, a P-glycoprotein transporter inhibitor, the toxic pump, is the interaction. Patient on digoxin is given clarithromycin. We already talked about a patient who has cardiac problem. He is on digoxin, a cardiotonic drug, and he has an infection, and so he receives clarithromycin prescribed by the physician. Guess what happens? Once again, I already gave a hint here, P-glycoprotein. So P-glycoprotein transporter is an important transporter present in the intestines as well as in the kidneys, the renal tubule. And so administration of clarithromycin, which is a very potent P-glycoprotein inhibitor, it will cause inhibition of digoxin efflux. That means digoxin throwing out is stopped, and so digoxin levels get accumulated in plasma, and this can result in digoxin toxicity. So either you change the antibiotic, that is, instead of using clarithromycin, you check the literature, you will see azithromycin is okay, it doesn't have this effect, or you cut down the dose of digoxin. I would rather prefer to go with the clarithromycin change, because it is difficult to quantitatively determine whether the effect is sufficient enough or not for reducing the bad effects of digoxin. The third one is CYP3A4 induction, and then tacrolimus, transplant patient has been taking tacrolimus. It is anti-rejection drug. He takes herbal St. John's wort. In the West, St. John's wort is a very famous herbal product like our ashwagandha. St. John's wort, is consumed by many people without doctors prescription. And this has multiple actions, like it inhibits the SSRI, it causes serotonin levels, increase in serotonin levels, dopamine levels, norepinephrine levels, and so on. And when you give St. John's wort, what it does is it can cause acute organ rejection. Why? This is because the St. John's wort it causes induction of CYP3A4. Induction means it increases the CYP3A4 enzyme levels. When enzyme levels are more, metabolism is faster or greater. And when metabolism is faster and greater, tacrolimus is very rapidly and greatly is decomposed. And as a result, you are having lower levels of active tacrolimus in the plasma. Therefore, the patient doesn't have adequate levels of tacrolimus to protect the transplanted organ like liver or kidney. Therefore, there is a chance of organ rejection. See, a simple interaction that could have been averted because the doctor doesn't know that the patient is taking a herbal product, but this can cause it. Likewise, there are other products. For example, grapefruit juice is another very important culprit perpetrator that inhibits several enzymes, particularly CYP3A4. And many doctors do not know that the patient has been consuming gallons of grapefruit juice. But they have to be told when they are on treatment with the drugs like tacrolimus, which are substrates of this particular enzyme. But since the food and other things, herbal products is not within the scope of today's discussion, I'm not talking much about that. So this is another patent. The patient who is on a statin like simvastatin or rosuvastatin, receiving cyclosporine, what would happen? So as I told already earlier, that OATP transporters are important transporters that are present in the liver, and they are gatekeepers for the entry of the statins into the liver for its pharmacological effect, at the same time for its metabolism. And once you administer a drug like cyclosporine that inhibits, or rifampicin that inhibits the OATP transporter, the statin cannot get into the liver, therefore its levels are increased. Once the levels in the plasma are increased, the outcome is rhabdomyolysis, that is increased muscle catabolism. So the victim is statin and the perpetrator is inhibitor that blocks the liver uptake at the door. So statin concentrations in the blood skyrocket, penetrating into the muscle, causing severe muscle pains and eventually rhabdomyolysis. So we need to, because these are included fortunately in the labels, and that is the best thing that has been happening now because of the proactive intervention through the guideline M12. You are able to capture all this information even before the drug is approved. So the prescriber's instructions will have all this information included in the label. I think this is the last interaction. The CYP2D6 and prodrugs. This is another very interesting. A breast cancer patient who has been on tamoxifen, you know pretty well that tamoxifen is a great drug for treating breast cancer. And the patient also has got depression, therefore receives paroxetine. For a different reason, the patient has hot flashes, the woman, and so she is receiving paroxetine. But what happens is there is no improvement in the breast cancer condition. The patient shows a therapeutic failure, cancer recurs. And again, this is a very serious condition, and unfortunately, the cause is a drug interaction. Tamoxifen, you know very well, is also like terfenadine, is a prodrug, and it is converted into endoxifen, an active molecule by CYP2D6. And paroxetine, we already discussed earlier, it causes inhibition of CYP2D6, and this can result in the inhibition of conversion of tamoxifen into active form. Therefore, the drug is ineffective, and that obviously would result in the failure in the therapeutic condition. So now look at this jungle. So this was a beautiful artistic image that was developed by the two guys, Louis Rocha and Rian Correa from the University of Indiana, Indianapolis. And this particular image shows that high risk drug interactions, the red lines, if you look at this picture, they are higher risk drug combinations among women. So the darker the color, the worse is the interaction. And the blue lines indicates the connections between higher risk drug interaction combinations among men. Typically cardiovascular drugs, CNS drugs, these are the ones which they stand pretty high in the line, and also certain gastrointestinal drugs, based on the color. This is only just to give you a glimpse as to how complicated that the drug interactions could be, or how common they could be in clinical practice, resulting in high risk outcomes. So I want to conclude by showing this slide. So we have got to be more predictive instead of being reactive. I already explained what this is. Instead of waiting all the way until the drug is approved for the reactions to be reported by the doctors in the clinical practice, you have got to be more predictive, making use of ICH guideline, where in vitro assessment, model-based assessment, and then, if required, a clinical-based assessment would guide us to understand the possible or potential drug interactions very early in the game. Then you can include those in the either the prescription, in the label, if it reaches that level, or the FDA may, or FDA or the regulatory agency may say that this drug has got a very strong interaction potential. Therefore, you have to include these in your label very clearly as probably black box warnings, or warnings and special contraindications, etc. So today we have been using a model informed drug development. Is called MIDD. FDA has been encouraging a lot using PBPK simulations, or using other virtual clinical trial related software. We can use even AI based models for understanding interaction potential very early in the process. And then precision management is that you are going to add pharmacogenomics also into the mix. I haven't touched anything about pharmacogenomics, which is very close to my heart. I always love to speak on pharmacogenomics. How this could influence or further confound the drug-drug interactions. And in the end, to everyone, to both clinicians as well as the regulators, is the apply 3M, that is monitor, modify, minimize. Monitor the interactions or therapeutic drug monitoring, modify wherever it is possible, and minimize through de-prescribing or reducing the prescriptions. I'll just show one. There are many like this. You can just Google, say, drug interaction checker, drug bank. It will take you to a website. And I'm not sure. Are you able to see this? Anyone can tell me? Are you able to see the screen?
Yes, sir.
Okay. Okay, so this is the screen. I will just type in one drug here. You can quickly check digoxin. Okay.
Sir,
so
we could able to see only PPT, not the website, sir.
Oh, okay. So in that case, I apologize. There is a link here, and I advise you to look into that. And otherwise, I could have shown you. You can readily understand. If there is a minute time, maybe I could switch to that one and share that screen to demonstrate. Otherwise, you can Google and see and ask for drug interaction checker drug bank. It will take you to the link directly. That is godrugbank.com, drug interaction checker. And this particular page will give you an option to use any combinations of drugs, and that will give you whether there is any interaction or not. If there is an interaction, whether that interaction is mild, moderate, or major, minor, moderate, or major. So that will give you some kind of ready reckoner kind of information that helps you. So thank you so much for giving your valuable time today. But one message I would like to give, that we all being pharmacists, or majority being pharmacists, of course, this is also applicable to all the clinicians who are on the call. So prepare our pharmacists, prepare our clinicians for the future challenges related to the drug-drug interactions. And I think it's time, if not to go to bed, at least to prepare to going for bed. Good night. Thank you so much.
Thank you very much, sir. And while going in this one, some important participants I see here are most of you are students only. Dr. Krishna Mohan Chinnala, APTI Telangana President, Dr. Ram Narasimha Reddy from Kakatiya University, Professor Sammaya Sir, Dean, and IPGA General Secretary, Professor Prabhakar Reddy Sir, from Palamur University, Dr. Jaga Shetty, former drug controller of Karnataka, and many more with PharmD students and other things. And I will be very happy if any one of these eminent persons can, if you message me, we will allow you to speak to sir, number one. And before that, sir, I have some few questions, maybe related to this topic, are also subjects related only. First of all, coming to the importance of pharmacy, what you are see the thing scenario in Europe or even Gulf countries, there is a lot of importance is given to the pharmacist there, sir. When I worked as a pharmacist, community pharmacist in Bahrain during 91-93 itself, if any drug which interactive or abnormal drug is given to a pregnant woman or a BP heart patient, we used to call the doctor, and the doctor used to give utmost respect to the pharmacist. Oh, I am very sorry, please send me back the prescription, we will correct it and send again, like that. Even US, you are well aware of about that. But coming to India, what happened nowadays, only little started with PharmD students, PharmD postgraduates are taking into the hospital pharmacy as a clinical pharmacologist and other things. Initially, they were rejected by the practitioners profession. I cannot, and most of them, they were expressing in front of us by calling, sir, we are getting insulted. And now slowly, because change has, first it will be rejected and later it will be think and then accepted, like that. Now people are little bit happy. Yes, sir, we are giving good feedback to the physicians and practicing doctors, and they are accepting slowly. It may take some more time. Because the role of PharmD definitely make a major role in such type of drug introductions and other things. Number one. Number two, the burning topic in India is paracetamol versus propylene glycol, sir.
Paracetamol and
Propylene glycol.
propylene glycol. Okay.
Propylene glycol, it started with that Cambia incident, sir, cough syrup, contaminated cough syrup deaths. From there, recently in India, also in Madhya Pradesh, it has happened. So the Drug Controller General of India and Government of India and state governments also very stringent in inspecting the cough syrup manufacturers, especially in the name of risk-based inspections, RBIs. So most of them are a small scale or medium scale companies are there, and the Government of India has a fixed date of 31st December 2025, the last date for complying the Schedule M. So my question is, in propylene glycol will be added in cough syrups when paracetamol is one of the ingredient. So paracetamol is water insoluble. That is the reason people are adding propylene glycol, which is sometimes people are adding commercial grade or propylene glycol without testing them for diethylene glycol and other impurities. That is the problem. This issue is coming. My final question is, why years together we are getting from, we started with the tetracycline, doxycycline, ampicillin, amoxicillin, cephalosporins, and so many antibiotics one by one, like that heterostatin level, so many this one. Why there is no alternative for years together you are using only Panadol or Tylenol or paracetamol only? Why not why not alternative drug is detected by our scientists for paracetamol for getting relief from the fever? Number two is why our scientists are failed in manufacturing a water-soluble paracetamol so that this hurdle may be overcome. Or else any other diluent which is safer than propylene glycol. Over to you, DRK sir.
Yeah, no. The first question, the answer is very simple: education. See, this, for example, today, I really commend your, conducting this particular seminar. I think, many people, must have been able to hear something related to the drug-drug interactions. And the same thing has to reach every practicing clinicians. In fact, one of the questions that I see here while scanning through the questions, somebody is talking about RMPs knowing. The RMPs is a very peculiar situation that is there only in countries like India, where their understanding about disease, understanding about treatments, understanding about drugs, drug interactions is such a complicated subject. And as I mentioned earlier, Lancet has very recently published an article where there is a codeine related issue. So like this, there are so many problems happening, and unless, the practicing doctors, are aware, and it is very difficult for the doctors to focus on all the drugs that are being prescribed. And so every hospital should have a responsible clinical pharmacist, not maybe many clinical pharmacists, and they should be taking enough responsibility related to the drug interactions and problems related to this kind of issues, adverse drug reaction monitoring. And then coming to your second question, ethylene glycol poisoning. Ethylene glycol poisoning is not new in the history. If you just scan through the literature, do you know when the first problem happened, poisoning occurred? In 1930, okay, where it was first observed, and many people died because that the product contained 95% ethylene glycol in the antifreeze. And in 1937, I think many of you are aware, this is a very classical example that happened in the US. 1937, the elixir of sulfanilamide, where it consisted of ethylene glycol. So it's not necessary that propylene glycol should have ethylene glycol. The problem is with the quality, and if there is no proper quality check that the propylene glycol is free from ethylene glycol, you cannot control this. Coming to paracetamol and water solubility, yes, paracetamol is not water soluble, I agree. But there are water-soluble paracetamol products. Our own company back in the US developed not just water-soluble paracetamol, it is water-soluble paracetamol injection. And paracetamol can be made water-soluble, but it is not necessary. So many drugs are water-insoluble. Therefore, we don't have to find, there are many other analgesics that are available, but paracetamol is still a great drug. So we don't have to find quickly another cheaper alternative for paracetamol. But as far as I know, the best way is to control the inactive ingredients. So the quality control is not about how much percentage of active ingredient is present in the final formulation, but we should also take care of how much amount of the approved inactive ingredients that are there. That is what we call GRAS substances or GRAS ingredients in the US FDA language. And so our DCGA also definitely prescribes the inactive ingredients that should be there and their quality. Go ahead.
Please mute all, please.
So that's about it. And there are a couple of questions which I would like to touch upon very quickly. Someone has suggested that whether RMPs are given knowledge of this or such many types of drug-drug interactions. So is it really possible? Even highly qualified physicians are not able to accept or grab information related to drug-drug interactions. And so education is the only key. Try to reach out to the rural practitioners, whether they are RMPs or even MBBS doctors who are practicing in smaller places where they do not have access to the information related to the drugs. So this is a wonderful platform Mr. Koteshwar and others, who have been doing tremendous service by organizing this kind of seminars, not just reaching out the pharmacists, but also reaching out the practicing physicians in the rural areas. Is important to give them greater knowledge, not only in terms of drug interactions, but in terms of drugs in general. What are the drugs, how they act, some information related to these could be definitely helpful. I think this question has been very aptly asked by Dr. Jagashetty. Great
Dr.
question.
Reddy, 1 minute, sir. I also involved with the answer to Dr. Sudarshan Reddy Dalchini, sir. These to meet this one, only continuous education is the only alternative. For that, we are ready to conduct a giant, trainings or special sessions to these RMPs. If the institutions are ready to come along with, join with us, APTA Association Pharmaceutical Teachers are ready. IPGA is ready, Drug Control Officers Association is ready. Only institutions should come forward. We are ready. You have to give us a platform so that in rural areas we can conduct direct physical meetings and educate them. This is the only alternative. Over to sir.
Absolutely, that's a good answer. And there is another question from another response from Dr. Sudarshan Reddy. In developed countries, they will check drug interactions using the checker. Absolutely, this drug interaction checker. I showed one. That is available. Online. Just Google it and ask for drug interaction checker. You have multiple sites where you can check drug interactions. People have been using smartphones every nook and corner of the villages even. So why can't a doctor or why can't a pharmacist just check whether there is an interaction between the two at the time of prescribing? I think, we have information right in our palms today, in our hands today. I think you only have to just go there, and now even apps are available. And one can check drug interaction potential, at least those interactions which are, major and moderate. And you also have plenty of tables that are provided by several universities, like University of, earlier I said, Indiana University. Likewise, FDA on their own website have got different drug interaction tables. This information is all over the place. The only thing is people should look for it. And in addition to that, this kind of workshops will give them, better education regarding, information related to the drug interactions.
Sir, one more question from Shivanjali, clinical pharmacist. For meropenem and sodium valproate interaction, apart from choosing other antimicrobial / anti-epileptic, any other management strategy? Sir, because is a culture directed therapy, meropenem for urosepsis and patient on valproate since ages.
Sorry, I'm not able to hear. Meropenem?
It is in meropenem and sodium valproate. It is in the messages, sir. Meropenem and sodium valproate interaction apart from choosing other antimicrobial or anti-epileptic. Any other management strategy? Sir, because a culture directed therapy, meropenem for urosepsis and patient on valproate since ages.
Meropenem, and the second one you said is valproate, right?
Sodium valproate, yeah.
Okay, sodium valproate. Okay, if you have the response, at your fingertips, as I said earlier, if you can just look into, there is an interaction between the two, right? Valproate and.
The question is not clear, sir. Next,
Dr. The question is not very clear.
Yeah. Dr. Jagashetty, former drug controller, Karnataka says, ciprofloxacin inhibits tizanidine is most important, but whether our RMPs are given knowledge, this is already answered.
Yeah, this I already mentioned. I think that's pretty much all. And if you still have any more questions, you are welcome to reach out to me via my email, dr_krishna@aol.com. Hello?
Yes.
Good evening, sir. Surya Kumar, yes, sir, student, sir. One thing,
Yeah.
can just comment on drug excipients interaction, sir, because that also is a big problem for the formulators.
That is what I have been telling, and we will have to check right in the development of the formulation. See, drug excipients enter the scenario because during the early drug development, the discovery followed by early drug development, which consists of in vitro and in vivo, assessment, non-clinical pharmacology. There, the focus is only on the active pharmaceutical ingredient, that is a new molecule, but not on the other additives. So the excipients, they enter the scenario at the time of formulation development. So at that point, there are a lot of interactions reported. For example, some of the ingredients that are used for enhancing the solubility of, the active molecule. I think many of you are aware that paclitaxel injectables contains, certain ingredients for making it more soluble, and those can cause a lot of issues. So these are to be evaluated, assessed, and then you should take them into regular practice of making the formulation.
Thank you, sir. Actually, sir has given only 1 hour time. We took 2 hours almost, 2 hours 10 minutes, and really wonderful session, sir. We are anticipating such events in future also, because since last 1 year I am just hunting you only because of our time factor. We could not able to conduct, and finally we did it, sir. And also give us some expert speakers in future also from US on our subject related matters. And now over to Dr. Parmanand Verma, Chairman, DCO, Drug Control Officers Welfare Association, Webinar Committee Chairman, to present his vote of thanks, please. And before that, if any students are there, kindly send me your details to my given WhatsApp number. I will try to send you participation certificates too. Thank you. Over to Dr. Parmanand Verma.
Thank you, Rao sir. Good evening, everyone. This is Dr. Parmanand Verma, here with the vote of thanks. It was a wonderful session and was very interactive. On behalf of DCO IWA, we are thankful to Dr. V. Kalaiselvan sir, the chief guest of today's session. Thank you very much, sir, for giving the insights of latest edition of Indian Pharmacopoeia. We are thankful to Professor Krishna R. Devarakonda sir for such a nice and elaborative presentation. Definitely, it will be beneficial for all of us. I am thankful to Honorable National President, DCO IWA, Mr. G.K. Rao sir, Mr. N.K. Ahuja sir, and my colleague, Mr. Krishna. And a special thanks to Mr. Uma. At last but not least, thanks to all participants for making this session successful. Thank you very much, sir.
Yeah. Requested all to stand for national anthem, please. Uma Shankar, go to.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Jai Hind and good night to all.